So we are jumping straight into the core of it today.
Straight into the deep end.
Yeah, because if you are listening to this right now, you are probably staring down the barrel of a massive biology syllabus. And right now we are zeroing in on a topic that it usually gets severely underestimated.
Oh, absolutely. Chapter 9.
Right, Chapter 9, Strategies for Enhancement in Food Production. It is very easy to look at that title and think, all right, agriculture, farming, crop yields, let's just memorize the bold words and move on.
That is the classic trap.
It is. But having a breakthrough moment with this material, taking a closer look at the actual progression of the text, it changes the entire perspective. We are not just looking at a dry list of farming techniques here.
No, we are looking at a survival guide.
Exactly. Humanity's ultimate survival guide against an explosive biological clock. This chapter is answering a singular, terrifying question. What happens when the biological carrying capacity of our planet is maxed out, but our population just keeps growing?
Taking that exact question and looking at how the text introduces it reveals the entire premise of the syllabus. When you spend over 15 years mentoring learners, particularly those preparing for highly competitive medical entrance exams, like Neet, you notice where the crucial disconnects happen.
Where did they usually miss it?
They skip the introductory paragraphs, they just want to get to the diagrams and the bold terms. But the source material lays out the stakes in the very first sentence.
What does it say?
It explicitly states, with ever-increasing population of the world, enhancement of food production is a major necessity.
Major necessity.
Right. The phrasing there is deliberate. It does not frame agricultural science as a passive study or just some economic venture. It frames it as a biological intervention. The ever-increasing population is the stressor. And the biological principles we apply are the necessary adaptation to ensure survival.
That concept of biological intervention, it requires a solid mental model for anyone studying it.
Absolutely.
So picture the world as a giant dining table.
Okay, I like this.
The guest list for this dinner party, the human population is doubling. People are continuously pouring into the dining room, pulling up chairs. But the physical size of the dining table, meaning the arable land on Earth, the actual soil we have to grow food in...
It is fixed.
It is fixed. It is not expanding. So what do we do when we cannot build a bigger table? We have to go into the kitchen and completely hack the way the food is being produced.
We have to change the fundamental rules of the kitchen's biological output.
Yes, so that the same amount of space produces 10 times the food. That is the essence of this chapter. It is about using biology to hack biology.
Extending that dining table analogy, the source material explicitly tells us the tools we are using to execute that hack. The text says biological principles, as applied to animal husbandry and plant breeding, have a major role in our efforts to increase food production.
Biological principles, that's the key phrase.
It is the pivotal concept for a learner to grasp immediately. We are taking the theoretical laws of genetics, the understanding of cellular mechanics, the raw rules of inheritance, and we are forcefully applying them to living systems.
We aren't building mechanical synthesizers to make food out of thin air.
Exactly. We are taking the exact same biological principles that govern our own cellular division and manipulating them in other organisms to force a higher yield. And to truly master this for an exam, you cannot just memorize isolated facts.
Right. You have to understand the architectural blueprint of how humanity has scaled these biological hacks over time.
Which perfectly sets up our mission for this deep dive.
Yeah. If you want to understand this topic, if you want to bulletproof your knowledge for the exam, you need a mental map. We are going to break this chapter down into 4 distinct pillars. It creates a logical flow that tracks humanity's technological evolution in food production.
The 4 main sections of the text.
Right. Section 9.1, Animal Husbandry, 9.2, Plant Breeding, 9.3 single-cell proteins, and 9.4 tissue culture. To lock this in, I use a framework, a mnemonic, basically. The APST pattern.
APST, let's break that down.
A for animal, P for plant, S for single-cell, T for tissue. It sounds simple, but it is actually a roadmap. A and P represent the macro-level foundation. S and T represent the micro-level frontier.
That APST pattern is an excellent cognitive anchor. It prevents the learner from seeing these 4 topics as isolated islands of information.
They're all connected.
Highly connected. Let us dive deeply into the macro-level foundation first, Section 9.1, Animal Husbandry.
The A.
Yes. When we talk about applying biological principles at this level, we're talking about manipulating entire multicellular complex organisms. For thousands of years, if humanity needed more food, we looked at the biggest, most visible variables we could control. We looked at the herd.
Right, the animals themselves.
But from a scientific standpoint, Animal husbandry is not just taking care of animals. It is the agricultural practice of breeding and raising livestock. It deals with the care and breeding of livestock like buffaloes, cows, pigs, horses, cattle, sheep, camels, and goats. And it extends further into poultry farming and fisheries.
Let's unpack the breeding aspect of that because that is where the actual biological hack happens. We aren't just letting the animals breed randomly in a field.
Not at all. We are actively intervening.
The text dives into the concept of a breed, which is defined as a group of animals related by descent and similar in most characters like general appearance, features, size, and configuration.
Right.
When we talk about breeding, we are essentially trying to concentrate the best biological traits into a single lineage. We are trying to build a better machine, but the machine is biological.
Precisely. And this is where the exam questions start to get highly specific.
They love testing them.
Oh, they do. In animal breeding, the examiner expects you to know exactly how we manipulate these genetic lineages. The most fundamental tool is inbreeding.
Mating of more closely related individuals.
Within the same breed for 4 to 6 generations. The biological principle here is straightforward. Inbreeding increases homozygosity.
Homozygosity.
Yes. If you want to create a pure line in any animal, just as Gregor Mendel did with his pea plants, inbreeding is strictly necessary. It exposes harmful recessive genes that can then be eliminated by selection, and it accumulates superior genes.
But there is a massive biological catch to leg that isn't there. You can't just inbreed forever.
Nature has limits.
Right. The biology eventually pushes back. This is where the concept of inbreeding depression comes in.
A very high-yield term.
If you keep mating closely related animals, eventually the fertility and even the productivity of the animal crash. The biological system gets too concentrated with those recessive alleles that might not have been fully purged, or it just loses the vitality that comes from a genetic variation.
That biological pushback is exactly what examiners focus on. When inbreeding depression occurs, the biological solution is a specific technique called outcrossing.
Outcrossing?
This is a crucial distinction for learners to memorize. Outcrossing is the practice of mating animals within the same breed. But, and this is the key constraint, they must have no common ancestors on either side of their pedigree up to 4 to 6 generations.
So same breed, but basically strangers genetically.
Exactly. A single outcross often helps to overcome inbreeding depression. It injects just enough genetic variance back into the lineage to restore fertility and yield while keeping the animal within the desired breed parameters.
So outcrossing is basically the reset button for inbreeding depression.
A perfect way to phrase it.
But what if we want to combine the best traits of 2 completely different breeds? Like, say we have one breed of cow that produces an enormous amount of milk, but it gets sick easily. And another breed is incredibly tough and disease-resistant, but doesn't produce much milk.
In that scenario, the biological principle applied is crossbreeding. You take the superior males of one breed and mate them with the superior females of another breed. This allows the desirable qualities of 2 different breeds to be combined.
And then what do we do with the offspring?
The progeny might be used for commercial production directly, or they might be subjected to some form of inbreeding and selection to develop a new stable breed.
Are there specific examples of this we need to know?
Absolutely. A classic example that frequently appears in exams is Hisardale.
Hisardale.
Yes, a new breed of sheep developed in Punjab. And you need to know exactly how it was developed. It was created by crossing the Canary ewes and Merino rams. Knowing these specific examples, the exact parentage of the cross is a non-negotiable requirement for the test.
The Canary ewes and Merino rams make Hisardale sheep.
Got it. It is fascinating because it is essentially genetic engineering done over generations using macroscopic animals.
It is.
And then there's interspecific hybridization, which takes it even further by crossing male and female animals of 2 different related species.
Right. 2 different species entirely.
The classic example is the mule bred from a horse and a donkey.
Oh.
You get an animal with immense physical stamina, but the biological trade-off is usually sterility. The biological principles of species boundaries kick in. That boundary is exactly why humanity had to develop more advanced techniques. Waiting for natural gestation, hoping the crossbreeding yields the exact genetic combination you want, it is incredibly slow.
Too slow for an ever-increasing population.
Precisely. The biological clock of a macroorganism like a cow is a bottleneck. This brings us to a highly critical concept in this Chapter 1 that the source material specifically highlights as having a pivotal role.
Here we go.
We are talking about embryo transfer technology, specifically multiple ovulation embryo transfer, or MOE.
MOE. This is where the kitchen hack becomes incredibly aggressive. If traditional breeding is just guiding nature, MOE is putting nature on an industrial assembly line.
That is a very accurate description.
Let's break down the biology of how this works because it perfectly illustrates the shift from basic farming to high-level biotechnology. Instead of waiting for a prize cow to produce one calf per year, we hack her reproductive system.
The mechanism of that hack is hormonal manipulation. In MOE, a cow is administered hormones with FSH-like activity.
FSH standing for follicle-stimulating hormone.
Correct. Normally, a cow's biological cycle induces the maturation and release of a single egg per cycle. By introducing synthetic or exogenous FSH, we induce follicular maturation and superovulation.
Superovulation.
Yes, instead of one egg, the cow produces 6 to 8 eggs in a single cycle.
Superovulation. That word alone tells you everything about the intensity of the intervention. So the cow produces 6 to 8 eggs. She is then artificially inseminated with the semen of an elite bull. Now she has 6 to 8 fertilized eggs embryos developing inside her, but we don't let her carry them to term because that would ruin the efficiency.
Exactly. The next step is where the technology truly bypasses the natural biological limits. The fertilized eggs, when they reach the 8 to 32 cell stage of embryonic development, are recovered.
8 to 32 cells.
Yes.
Yeah.
Memorize that range. And crucially, this recovery is done non-surgically. The embryos are flushed out of the donor cow's reproductive tract and then transferred into surrogate mothers.
This is the genius part. The genetic mother, the elite cow with the superior genes, is completely freed up.
She is back to square one.
Right, she doesn't have to spend 9 months carrying a calf. She can be superovulated again almost immediately. Meanwhile, the surrogate cows, who might be totally average genetically, are serving as the biological incubators.
Yes.
You can take the genetics of one prize cow and produce dozens of her offspring in a single year using an army of surrogates.
And that is why the introductory text uses the phrase pivotal role. MOET has been successfully demonstrated in cattle, sheep, rabbits, buffaloes, and mares. High milk-yielding breeds of females and high-quality meat-yielding bulls have been bred successfully to increase herd size in a fraction of the normal biological time.
It's incredible.
But there's a massive trap here for students reading the introduction. When they read the phrase enhancement of food production, their minds immediately default to plants, to agriculture, to tractors and seeds.
Oh, the plant bias.
The plant bias.
It is so easy to fall into. When we hear food production, we think of wheat fields, not embryo flushing in cattle.
Exactly. Examiners are acutely aware of this cognitive bias. They know that students gloss over the animal side of the equation. So when a multiple-choice question asks which new techniques are highlighted as playing a pivotal role in further enhancing food production, and one of the options is embryo transfer technology, thousands of students will eliminate it.
Because they falsely assume food production only means plant breeding.
Yes. You must explicitly link animal husbandry and embryo transfer technology to the core concept of solving the human population crisis.
So if you are studying this, draw a massive red line connecting MOE and embryo transfer directly to the overarching goal of food enhancement. Do not let your brain trick you into thinking this chapter is only about green things.
Very well said.
But speaking of green things, let's move to the second pillar of our APST framework, the P plant breeding. Section 9.2. If animal husbandry was our first macro hack, plant breeding is the hack that actually saved the modern world from starvation.
It is impossible to overstate the importance of this section. When we discuss plant breeding, we are inherently discussing the Green Revolution.
The Green Revolution.
By the mid-20th century, the global population was accelerating at a rate that traditional agriculture simply could not support. Traditional farming varieties were hitting their absolute biological ceilings in terms of yield. Plant breeding as a technology was the only viable intervention.
How does the test define it?
It defines it as the purposeful manipulation of plant species in order to create desired plant types that are better suited for cultivation, give better yields, and are disease resistant.
And just like with the animals, this isn't just planting seeds and hoping for the best. It is a rigorous, systemic, multi-step scientific process. Let's walk through the actual biological steps of creating a new genetic variety of a crop, because this is prime testing material.
They will test the sequence.
They always do. So it starts with the absolute foundation of all biology, genetic variation.
Step 1 is the collection of variability. Genetic variability is the root of any breeding program. In many crops, pre-existing genetic variability is available from wild relatives of the crop.
So they go hunting for wild plants.
Yes. The entire collection of plants and seeds having all the diverse alleles for all genes in a given crop is called germplasm collection.
Germplasm collection.
This is your raw material. If you do not have a robust germplasm, you have nothing to breed.
Think of it like having a library of every possible genetic trait. Once you have the library, you move to step 2. Evaluation and selection of parents. You look at this massive collection and you identify the plants with the desirable combination of characters.
Give me an example.
Okay, you want a wheat plant that has a short, sturdy stalk so it doesn't fall over in the wind, but it also needs to produce massive amounts of grain. You select those specific parents and you multiply them, often creating purer lines through inbreeding, just like we discussed with the animals.
Then comes step 3, which is where the intensive labor occurs.
Yeah.
Cross-hybridization among the selected parents.
This is the physical combination.
Yes, this is where you physically combine the genetics. If parent A has high protein content and parent B has disease resistance, you must cross them to combine these traits in the progeny. Biologically, this is a tedious and time-consuming process.
Because you're doing it by hand, right?
Hand pollination. It involves emasculation, which is the careful removal of anthers from the bisexual flower of the female parent before the pollen matures. And then bagging, which is covering the emasculated flower to prevent contamination by unwanted pollen. Then you manually collect pollen from the male parent and dust it onto the stigma of the female parent.
And the staggering part is the statistical probability of success. You can do this precise, delicate hybridization thousands of times, and the resulting progeny might not have the combined traits you want. It's not a one-to-one guarantee.
It rarely is.
It is a numbers game played on a massive scale.
Which necessitates step 4, selection and testing of superior recombinants. You have to grow out the thousands of hybrid progeny and meticulously select the ones that actually exhibit the desired combination of characters.
And once you find that perfect plant,
these plants are then self-pollinated for several generations until they reach a state of homozygosity, meaning the traits are fixed and the progeny will not segregate in further generations.
so it becomes a pure, predictable line.
Exactly. Finally, step 5 is testing, release, and commercialization of new cultivars. The newly selected lines are evaluated for their yield and agronomic traits by growing them in research fields, recording their performance under ideal fertilizer application, irrigation, and crop management, and then testing them in farmers' fields across different agroclimatic zones.
The sheer scale of that operation is mind-bending, and the results changed history. In 1960, India was facing massive agricultural shortfalls. But through these exact plant breeding techniques focusing on semi-dwarf varieties of wheat and rice, the production numbers exploded.
The numbers are historic.
Wheat production went from 11 million tons to 75 million tons. That is the biological intervention in action. That is the dining table being hacked to produce exponentially more food from the exact same acreage of land.
The individuals who pioneered this, like Norman Borlaug, who developed semi-dwarf wheat at the International Center for Wheat and Maize Improvement in Mexico, fundamentally altered human carrying capacity.
They saved billions.
Literally. In India, derivatives like Sonalika and Cayenne Sona were introduced, which were high-yielding and disease-resistant.
Sonalika and Cayenne for wheat.
Correct. For rice, semi-dwarf varieties were derived from IR8, developed at the International Rice Research Institute in the Philippines, and Taichung Native 1 from Taiwan. Later, even better-yielding semi-dwarf varieties like Jaya and Ratna were developed in India.
Jaya and Ratna.
These specific names, Sonalika, Cayenne Sona, IR8, Jaya, Ratna, they are high-yield facts for any biology exam. They will ask you to match the variety to the crop.
But plant breeding isn't just about quantity, right? It isn't just about making a bigger pile of grain. It's also about fighting the biological threats that try to destroy the food before we can eat it. We are talking about disease resistance.
A constant battle.
Right. A wide range of fungal, bacterial, and viral pathogens affect the yield of cultivated crop species.
Absolutely. If a crop is susceptible to rusts, smuts, or blights, the yield can be entirely decimated, rendering the high-yield genetics useless. Therefore, breeding for disease resistance is a parallel necessity.
How does that work genetically?
The biological principle is to identify resistance genes in the germplasm or in wild relatives and hybridize them into the high-yielding varieties. For example, the wheat variety, Himgiris, is bred for resistance to leaf and stripe rust.
Himgiri for wheat rust.
And the brassica variety, Pusaswarnum, is resistant to white rust. These are classic examples of genetic fortification against environmental stressors.
And there is another layer to this fortification, which I
Yeah.
find incredibly elegant. It's not just fighting diseases that attack the plant, it's fighting diseases that attack us, the humans, eating the plant.
Ah,
This
yes.
is the concept of biofortification. We realize that just providing calories isn't enough. If the population is surviving on massive amounts of rice, but that rice has no vitamin A, you get widespread malnutrition, blindness, and immune deficiency. This is what they call hidden hunger.
Hidden hunger is a critical term. Over 3 billion people suffer from micronutrient protein and vitamin deficiencies because they cannot afford to buy adequate fruits, vegetables, legumes, and meat. Diets lacking essential micronutrients, particularly iron, vitamin A, iodine, and zinc, increase the risk of disease, reduce lifespan, and reduce mental abilities.
It's devastating.
It is. Biofortification is the strategic breeding of crops with higher levels of vitamins and minerals or higher protein and healthier fats. It is the most practical means to improve public health.
It's upgrading the nutritional density of the crop itself. Imagine instead of handing out vitamin pills, you engineer the daily bread to have the vitamins built in.
Precisely.
In 2000, they developed maize hybrids that had twice the amount of the amino acids lysine and tryptophan compared to existing maize hybrids. They created the wheat variety Atlas 66, which has a significantly high protein content.
Atlas 66.
Yeah, they've bred iron-fortified rice containing over 5 times as much iron as commonly consumed varieties. The Indian Agricultural Research Institute in New Delhi has released vitamin A-enriched carrots, spinach, and pumpkins, and vitamin C-enriched bitter gourd, bantha mustard, and tomato.
The detail of those examples is precisely what an examiner will test. They will ask you to identify Atlas 66, or they will ask what specific amino acids were doubled in the biofortified maize, lysine and tryptophan.
They love the specifics.
They do. But looking at the broader narrative, we must recognize a fundamental limitation. Despite the incredible successes of the Green Revolution, despite biofortification, despite MOET and animal crossbreeding, we eventually hit a thermodynamic wall.
Right. And this brings us to the crucial pivot in our mental map. We've covered A&P, animal husbandry, and plant breeding, the macro level, but as the introductory text warned us, the population is ever increasing. We are optimizing the macroorganisms as much as biologically possible, but a cow still takes months to grow.
Years sometimes.
Exactly. A wheat field still requires vast acreage, soil, water, and an entire season to mature. The macro clocks of nature are too slow, and the spatial requirements are too large. We need a new dimension to expand into.
Which necessitates the shift to the micro level. This is where the source material transitions from traditional agricultural science into the realm of modern biotechnology. We move to the S in our APST pattern, section 9.3, single-cell proteins.
Single-cell proteins.
The conceptual leap here is staggering.
Wow.
We are abandoning the multicellular organism entirely. We are no longer dealing with roots, stems, leaves, bones, or organs. We're looking at the thermodynamic inefficiency of the traditional food chain and choosing to bypass it completely.
Let's break down that thermodynamic inefficiency because it is mind-blowing. The shift from a grain-based diet to a meat-heavy diet creates an exponential demand for cereals. Why? Because it takes anywhere from 3 to 10 kilograms of grain to produce just 1 kilogram of meat by animal farming.
3 to 10 kilograms.
Think about that. You are feeding a cow 10 kilos of perfectly good calories, and the cow burns most of it just walking around breathing and keeping warm, yielding only 1 kilo of meat for us. It is an incredibly inefficient biological conversion rate.
That inefficiency is why more than 25% of the human population is suffering from hunger and malnutrition. Conventional agricultural production of cereals, pulses, vegetables, and conventional animal husbandry cannot meet the demand at the rate the population is scaling. Single-cell protein, or SCP, is one of the alternate sources of protein for animal and human nutrition.
How does it work?
It involves growing microbes on an industrial scale as a source of good protein.
This is where we talk about microbes like spirulina. Imagine this. Instead of needing hundreds of acres of prime, fertile farmland to grow soybeans for protein, you can take wastewater from a potato processing plant.
Water that is full of starch.
Exactly. Water full of starch, and use it as a nutrient broth to grow tons of spirulina, a blue-green alga. You can grow it on straw, on molasses, on animal manure, and even on sewage.
The elegance of spirulina is twofold. First, it produces massive quantities of food rich in protein, minerals, fats, carbohydrates, and vitamins. Second, because it utilizes waste products as a growth medium, it simultaneously reduces environmental pollution.
It is a closed-loop biological system.
It is. But to truly understand why SCP is considered a frontier technology, you have to look at the mathematical scale of microbial biomass production compared to macroorganisms.
The math is where the whoa moment happens for me. Let's compare a macroorganism, a cow, to a microorganism.
Let's hear it.
You have a 250-kilogram cow, a massive animal.
Yeah.
Every single day, that 250-kilogram cow produces about 200 grams of protein. That is its biological limit.
200 grams.
Now take a microorganism, specifically a bacterium like Methylophilus methylotrophus. Because of its incredibly high rate of biomass production and cellular growth, if you cultivate 250 grams of that bacteria, just a tiny handful of microbial mass, it can be expected to produce 25 tons of protein in a single day.
25 tons.
25 tons of protein from a 250-gram starting culture compared to 200 grams from a 250-kilogram cow.
The exponential difference in yield is exactly why the text emphasizes these new techniques. You are no longer bound by the macroscopic constraints of agriculture. You are operating on the micro clock of cellular division, which is exponentially faster. It has been calculated that microbes, like Methylophilus methylotrophus, represent a quantum leap in biological efficiency.
It's almost hard to comprehend.
And furthermore, the fact that edible mushrooms are now widely cultivated and consumed on a large scale proves that macroscopic fungi, which are conceptually related in terms of bypassing traditional photosynthesis-based agriculture, are a highly viable component of the human diet.
It is essentially 3D printing nutritional value at the cellular level, but as amazing as single-cell proteins are, they are mostly providing raw nutritional bulk. They aren't replacing the structure of the plants we rely on. We still need the physical plants, but we need to produce them faster, cleaner, and in massive quantities without needing massive amounts of land to start the process.
Which brings us to the final frontier.
The final and perhaps most futuristic pillar of our mental map. The T in APST, section 9.4, tissue culture.
Tissue culture is the ultimate manifestation of understanding biological principles at the cellular level. While traditional plant breeding relies on seeds and natural germination cycles, tissue culture allows us to propagate entire fields of plants, starting from just a microscopic cluster of cells in a laboratory.
In a lab.
Yes. During the 1950s, scientists made a profound biological discovery. They learned that whole plants could be regenerated from explants.
Let's define explant, because that is a high-yield vocabulary word. An explant is simply any part of a plant that is taken out and grown in a test tube under sterile conditions in a special nutrient media. It could be a piece of a leaf, a piece of a stem, or just a few cells from the growing tip of the plant.
And the biological principle that makes this possible is called totipotency. This is perhaps the most critical concept in this entire section.
Totipotency.
Totipotency is the inherent capacity of a single plant cell to generate a whole complex plant. In humans, only our very early embryonic cells are truly totipotent. But in plants, many mature cells retain this genetic blueprint and the ability to restart the developmental process from scratch, provided they are given the correct chemical signals.
The correct chemical signals. That is the nutrient media. You can't just put an explant in tap water. You have to provide the exact chemical environment the cell needs to divide and differentiate. This nutrient medium must provide a carbon source, such as sucrose, and it must contain inorganic salts, vitamins, amino acids, and crucially, growth regulators.
Those growth regulators are the steering wheel of tissue culture. The exam will frequently test your knowledge of these regulators, specifically auxins and cytokinins.
Auxins and cytokinins.
By manipulating the ratio of auxins to cytokinins in the nutrient medium, scientists can dictate exactly what the explant does. A high ratio of auxin promotes root development. A high ratio of cytokinin promotes shoot development. You are chemically dictating the biological architecture of the new plant.
Think of it like a biological remote control. And because you are doing this in a test tube, you aren't waiting for a season to change. You can take one plant, chop it into thousands of tiny explants, put them all in nutrient media, and within a very short period, you have thousands of plantlets ready to be transferred to soil.
Exactly.
This method of producing thousands of plants through tissue culture is called micropropagation.
And micropropagation has a massive commercial advantage. Each of these thousands of plants will be genetically identical to the original plant from which they were grown. They are clones. In biological terms, they're called somaclones.
Somaclones.
Yes. If you have a single highly elite variety of tomato, banana, or apple that has perfect yield and taste, you don't want to risk losing those traits through the genetic shuffling of traditional seed reproduction. Micropropagation guarantees that every single one of the thousands of offspring is a perfect genetic copy. Many important food plants like tomato, banana, and apple have been produced on a commercial scale using this method.
But the magic of tissue culture doesn't stop at mass production. It also acts as a biological cleansing mechanism. Let's say you have that elite perfect variety of potato, but the entire crop gets infected with a systemic virus. In traditional farming, that lineage is dead. You have to burn the crop to stop the spread. But with tissue culture, you can actually rescue the genetics from the dying infected plant.
This is the application of meristem culture, another highly testable concept. Even if a plant is severely infected with a virus, the meristem, which is the rapidly dividing tissue at the apical and axillary growing tips of the plant, is generally free of the virus.
Why is it free of the virus?
The biological principle here is that the cellular division in the meristem outpaces the viral replication, or the vascular system hasn't fully developed to transport the virus into that specific zone.
So you take a microscopic scalpel, remove just the meristem from the infected plant, and grow it in vitro as an explant. Because the meristem is clean, the new plant that grows from it will be completely virus-free, even though its parent was dying of the infection.
It's incredible. Yeah.
Scientists have successfully cultured meristems of banana, sugarcane, and potato to salvage elite lineages. It is essentially using a microscopic biological loophole to outmaneuver a pathogen.
And if we push the boundaries of tissue culture even further, we arrive at the absolute frontier of genetic manipulation without transgenic modification. Somatic hybridization.
This is where things get wild.
Very wild. In traditional plant breeding, as we discussed, you can only crossbreed plants that are sexually compatible. A wheat plant can only cross with another wheat plant or a very close wild relative. But what if you wanted to combine the traits of two completely different sexually incompatible plants?
This is straight out of science fiction. Somatic hybridization bypasses sexual reproduction entirely. It happens at the cellular level. First, scientists take a single cell from plant A and a single cell from plant B. But plant cells have rigid cell walls. You can't just smash them together.
No, they would just bounce off each other.
Right, so you use enzymes, specifically pectinase and cellulase, to digest the cell walls. What you are left with is the naked cell bounded only by its plasma membrane. This naked cell is called a protoplast.
Protoplast isolation is the crucial first step. Once you have isolated the protoplast from two different varieties of plants, each having a desirable character, you can chemically or electrically induce them to fuse together. When they fuse, their cytoplasm and the nuclei combine, creating a single hybrid cell that contains the genetic material of both distinct parent plants.
It's a completely new cell.
This is a somatic hybrid, and the process is somatic hybridization.
And because of the biological principle of totipotency, you can take that single-fused somatic hybrid cell, put it in the right nutrient media, and grow an entirely new complex plant that never existed in nature. The most famous example of this is the tomato. Scientists actually fused a protoplast of a potato with the protoplast of a tomato. They grew it into a full plant.
Now, it is important to note for the exam that while the tomato was a profound biological success, it did not have all the desired combination of characteristics for commercial utilization.
It didn't taste very good.
No, it did not. It proved the concept, but it wasn't the agricultural silver bullet they hoped for. Nevertheless, somatic hybridization remains a powerful tool in the biological arsenal, allowing for genetic combinations that traditional macrobreeding could never achieve.
Okay, we have covered an immense amount of ground. We have mapped out the entire APST framework: animal husbandry, plant breeding, single-cell proteins, and tissue culture. We have seen how humanity moved from selectively breeding massive herds over generations to chemically fusing naked protoplasts in a test tube.
A massive journey.
The scale of the shift is breathtaking. But for the learner listening right now, the student staring down the Neet exam, how do they actually synthesize this? Let's transition into the specific exam strategy, the Neet focus.
Synthesizing this requires understanding how examiners construct their questions. In a multiple-choice format, the incorrect options are rarely random. They are carefully designed traps based on common student misconceptions.
Right.
We have already discussed the plant bias trap regarding food production and embryo transfer. Another major trap involves confusing the terminology within tissue culture.
Yeah, they will mix up words that sound similar or belong to the same process but mean entirely different things. For example, confusing an explant with a somaclone.
Precisely. A question might ask, what is the term for the genetically identical plants produced through micropropagation? The options will be A, explants, B, somatic hybrids, C, somaclones, D, protoplasts. If a student only has a surface-level understanding, all of those words sound like tissue culture science words.
But an astute learner knows that an explant is the starting material. A protoplast is a cell without a wall. A somatic hybrid is the fusion of two protoplasts, and only the somaclone is the genetically identical end product of micropropagation.
You have to know the exact biological sequence. Explant goes into media, grows into a plant via totipotency, and those thousands of identical plants are somaclones. Another trap I can see is regarding the nutrient media itself. They could ask what is required in a tissue culture medium and offer options that miss the growth regulators.
Yes, they will test your knowledge of auxins and cytokinins. They might frame it as a scenario. A scientist wants to induce rapid shoot formation in a callus culture. Which growth regulator ratio should be applied?
And if you don't know that cytokinins drive shoot development while auxins drive root development, you will guess incorrectly. The exam rewards precision.
And precision is key when looking at the examples of biofortification and traditional breeding. Atlas 66 is wheat with high protein. Himgiri is wheat with rust resistance. Pusa swarnum is brassica with white rust resistance. You cannot mix those up. Golden rice or biofortified rice is about iron or vitamin A. You have to anchor the specific crop to its specific biological enhancement.
To solidify all of this, let us lock in the memory anchors. I want every listener to explicitly focus right now. Here is what you must remember. I'm going to give you the 5 critical facts checklist. These are the non-negotiable conceptual pillars of Chapter 9.
I'm ready. Fact number 1.
Fact number 1. The foundational driving force behind every strategy in this chapter is the ever-increasing population of the world. This is not a chapter about economic optimization. It is about mitigating a biological carrying capacity crisis. If an exam asks for the primary motivation for these enhancements, it is population pressure.
The dining table is running out of room. Fact number 2.
Fact number 2. The core mechanism utilized across all these diverse fields, from dairy farming to protoplast fusion, is the application of biological principles. We are not defying nature. We are weaponizing its own rules, like homozygosity, totipotency, and superovulation to force higher yields.
Biological principles are the tools in our kitchen. Fact number 3.
Fact number 3. In traditional breeding, you must distinguish between the solutions for different problems. Outcrossing is the specific biological remedy for inbreeding depression in animals. It introduces variation without losing the breed identity.
Outcrossing resets the depression. Fact number 4.
Fact number 4. Do not fall for the plant bias. Embryo transfer technology, specifically MOE, is explicitly highlighted as a pivotal new technique for food enhancement. It involves FSH-like hormones, superovulation of 6 to 8 eggs, and non-surgical recovery at the 8 to 32 cell stage. It is animal husbandry on an industrial biological scale.
MOE hacks the gestational clock. And the final fact, fact number 5.
Fact number 5. Tissue culture relies on the principle of totipotency. You must know that meristem culture is used to salvage virus-free plants, and somatic hybridization involves fusing naked protoplasts using pectinase and cellulase to create entirely new genetic combinations like the pomato.
That checklist is an absolute fortress of knowledge. If you layer those 5 critical facts over our APST structural map, animal-plant single-cell tissue, you essentially have a photographic conceptual memory of the entire source material.
That is the ultimate goal. When you sit down for the Neet exam or any high-stakes evaluation, you shouldn't be desperately searching your memory for isolated facts. You should be navigating a logical framework that you deeply understand. You aren't just reading. You have decoded the biological narrative.
It changes the way you look at a textbook. It's not a chore. It's a history of how we engineered our own survival.
All right, we have gone incredibly deep today. Let's bring it all together for the listener. Here is your rapid-fire 60-second sticky summary. Are you ready?
Let's hear it. The world is getting dangerously crowded. Our biological carrying capacity is maxed out. To survive, we took the fundamental rules of life, biological principles, and forcefully applied them to our food. We started macro with the A&P of our APST pattern, animal husbandry, and plant breeding. We used inbreeding, crossbreeding, and the Green Revolution to scale up whole organisms, fighting inbreeding depression with outcrossing and fighting diseases with targeted genetics and biofortification. But the macro clocks were too slow and land is finite, so we zoomed in. We shifted to the micro, the S&T single-cell proteins in tissue culture. We bypassed the inefficient cow by growing tons of spirulina on wastewater. We bypassed the field by using totipotency to grow thousands of identical somaclones from microscopic explants in test tubes, and we fused naked protoplast to create hybrid plants that nature never intended. We moved from farming the land to hacking the cell. Boom! That is Chapter 9.
And an exceptional synthesis. You capture the historical progression, the biological mechanisms, and the sheer urgency of the topic perfectly. But before we sign off, I want to leave our listener with one final provocative thought to mull over.
Ooh, okay.
We have established that tissue culture and single-cell proteins represent our current ultimate frontier, our micro-level response to an ever-increasing population. We have pushed biological principles down to the level of the single cell. But this raises a profound question. The population keeps growing. What happens when even those microscopic biological principles hit their absolute thermodynamic limit?
Wow.
When a cell simply cannot divide any faster, or be forced to produce even a fraction of a gram more protein, when the biological hack is fully optimized and the dining table is still too small, what could possibly be the next chapter in humanity's survival guide?
Oh, wow. That is a terrifying but brilliant question. It really highlights that science is never a finished book. It is a constant, desperate race against our own success. That is definitely something to chew on, pun entirely intended. Thank you so much for joining us and unpacking this massive topic. To you listening, keep questioning the biological systems around you. Do not just memorize textbooks, interrogate them. Find the survival story hidden in the structure. We will catch you on the next deep dive.